So hello everyone, my name is Pontus Forsell. I'm head of discovery and research here at Alzecure Pharma. Also one of the co-founders and preclinical project lead of the TrkA-NAM program. Today I will present a presentation entitled Next Generation Non-Opioid Analgesics. I will just give you a short background to the target pathway. So upon a release in the body, it could be as depicted here in the skin or in a deeper lying tissue like the knee joint. NGF is released from surrounding cells at the trauma site. This released NGF then travels to different kinds of cells where the receptor for NGF is expressed, and the receptor is called TrkA. So those cells could be immune cells like lymphocytes, macrophages, or resident mast cells in the skin. So there is a non-neuronal effect of NGF TrkA leading to increased inflammation at the site of the injury. NGF also binds to TrkA receptors on neuronal cells, as depicted here in this nerve ending in this figure here. So upon binding of NGF to TrkA, the nerve fibers will be sensitized. They will upregulate certain ion channels like TRPV1, NaV1.7, and NaV1.8, leading to allodynia at the site of injury and also a pain sensation. So this NGF TrkA pathway is very well validated. In man, it's actually validated by either deletion or mutations of the NGF or TrkA gene. These mutations or deletions lead to a painless phenotype. So these individuals do not sense pain. And that is, of course, not good because pain is the human body's defense. So you will not use the parts of the body that has been injured. And secondly, the pathway has also been validated by the anti-NGF antibodies, which actually demonstrated a step change in clinical efficacy in treating several types of pain. So to summarize this pathway, NGF TrkA pathway is very well validated both within nociceptive and neuropathic pain. So we'll talk a little bit about more about the pathway and especially the neurotrophins, which is a family of protein hormones which NGF belongs to. So NGF stands for nerve growth factor, and it's one of the neurotrophins, as you can see here. There are other neurotrophins named BDNF for neurotrophin 4 and 5 and NT3, and they also come in pro form. So we have a pro form of pro-NGF or pro-BDNF. And these different neurotrophins binds to different receptors. So I can see in this figure here, NGF binds to TrkA, and the other neurotrophins bind to specific other receptors on the cell membrane. And the receptor to the right here is called P75NTR. It's a common receptor for all the neurotrophins and all their proforms. So it doesn't only bind NGF, but it also binds NT4 and NT3. The different receptors have different functionality, of course, but the TrkA, B, and C receptors signal in similar pathways. So that's probably spatial or temporal dependent. The P75 receptor has a totally different signaling pathway, more regulating cellular survival or apoptosis. So several attempts has been made to target this pathway for the treatment of pain. And the first attempt involved general inhibitors of both TrkA, TrkB, and TrkC. So these are called TrkA inhibitors. So they target the catalytic site of the receptor, which is a kinase domain. They are non-selective, so they inhibit all three different forms of the TrkA receptor, but not the P75 receptor. So these molecules were discontinued for the treatment of pain due to severe adverse events like CNS-related side effects. There are now two different compounds in clinical use for the treatment of different malignancies. So the second generation was the anti-NGF antibodies. So they are delivered by infusion, so not by a tablet or pill, and they neutralize NGF or pro-NGF, thereby inhibiting the binding of the ligand to the receptor. So the anti-NGF antibodies will inhibit both the P75 signaling as well as the TrkA signaling. So they are more selective than the TrkA inhibitors, but still the anti-NGF antibodies do inhibit two different pathways, the P75 receptor-mediated pathway and the TrkA mediated pathway. So the third attempt to target this pathway involves the negative allosteric modulators, of which ACD137 belongs to. So this is a more, and actually the most selective mechanism of targeting this NGF-TrkA pathway. These molecules only bind to TrkA, so they are highly selective towards TrkB and TrkC, and does not at all affect P75 signaling. And currently we are only aware of two different negative allosteric modulators of TrkA that is under development, and that is ACD137 and a compound that is in phase 2 right now for the treatment of over pain by Alzecure. So to summarize this, TrkA is a validated pathway for the treatment of pain. Allosteric modulators might give opportunity also for biased signaling because they are more modulating the activity rather than inhibiting it. This slide is just a summary showing different indications that the anti-NGF antibodies were being developed for. So these are 3 different antibodies, tanezumab, furlanimab, and fasinumab from Pfizer, Lilly, J&J, and Regeneron. They are either in phase 3 or in phase 2, and many of these studies showed a positive effect on the pain perception. So it gives a flavor of other potential indications that molecules targeting the NGF-TrkA pathway can be used in. And here we can see also different forms of peripheral or neuropathic pain, like diabetic peripheral neuropathy or cancer pain or post-herpetic neuralgia. However, these anti-NGF antibodies suffered some severe adverse events like RPOA, rapidly progressing OA, found in a small portion of patients, but still enough to be dose limiting for the anti-NGF antibodies. So why is that? Why did we see this effect of the anti-NGF antibodies? Well, our hypothesis is that it's due to the fact that the anti-NGF antibodies are not only inhibiting the NGF-TrkA pathway, but also the pro-NGF or NGF pathway mediated by P75, the P75 receptor. There are now quite many reports describing the involvement of P75 in bone mineralization or in bone homeostasis, suggesting that there is a link between P75 and potentially rapidly progressing OA also. It was also a nice paper in Nature a couple of years ago where they demonstrated that P75 actually limits the inflammation in the knee joint in an arthritis model in animals. So removing this brake by inhibiting the interaction between NGF and P75, which the anti-NGF antibodies do, will unleash that brake, and you get an escalation of the inflammation. Also, the TrkA inhibitors that are on the market for oncology, like I mentioned, it's been given to a couple of thousand patients right now. It's larotrectinib and another molecule called entrectinib. These molecules have so far not been reported to have anything resembling RPOA as an adverse events. And finally, TrkA-NAMs have been demonstrated to have improved CNS safety profile. And in the bottom of the picture here, you can see an article or a figure from an article demonstrating what happens to the femur, which is lårben in svenska, And this is the cortical bone, so the dense outer layer of the bone. And if you knock out P75 from these animals, you can see that the density of the bone is much less than in wild types animals. And it also is similar for the trabecular bone, the more soft bone within the inside of the actual bone. So that sort of wraps up the background of this program. Now I will go a little bit into the actual TrkA-NAM program that they have here at Alzecure. So it's actually a program that goes back more than 10 years in the company. It was based on positive allosteric modulators of TrkA receptors that were identified early in 2013. So within this chemical series, we then, after a couple of years, identified selective negative allosteric modulators. So they were inhibiting the TrkA activity rather than increasing it as with the PAMs. Now, myself and our Director of Medicinal Chemistry, Gunnar Nordwall, we have worked on this target at previous positions in other companies, and we knew that identifying molecules with this kind of profile was not easy. So we decided to embark on a journey where we optimize this initial hit. And you can see in the figure to the right here that this initial hit is actually the black line to the right. And after lead optimization of this molecule, we were able to increase the potency of the molecules very much, and at the same time retaining a good selectivity towards other TrkA receptors. And ACD137 was selected as a clinical candidate in 2024. So this is a slide showing just a summary of the TrkA-NAM program. Like I mentioned, we had a quite extensive lead optimization effort. We synthesized more than 860 molecules. We were able to identify or synthesize molecules with very good potency, below 10 picomolar, which is extremely potent molecules. In general, they have very good selectivity towards TrkB and TrkC, usually higher than 10,000-fold, and also solve the X-ray structure of the molecule. And importantly, they have, in general, low blood-brain barrier permeability, so meaning that the molecule will not be exposed to such a high levels in the brain, which is good from a CNS-related side effects. And we have finally demonstrated that several TrkA-NAM molecules have analgesic and anti-inflammatory effects in various in vivo models of pain. So why do we think that selectivity for TrkA over TrkB and TrkC is important? Well, all the TrkA receptors are involved in neuronal function, both during the development, but also in the adult individuals. So it's important from a CNS side effects, but also from side effects related to peripheral nervous system that you are not targeting TrkB and TrkC. And this is a summary of different molecules, different TrkA-NAM molecules, also two TrkA inhibitors, as well as an anti-NEF antibody here. Tanezumab is included in the table. And I just want to highlight ACD137 in the top row, where you can see we have an IC50 on TrkA receptor, approximately 1 nanomolar, and it's 20,000 nanomolar, more or less, on TrkB and TrkC. So given it's roughly 20,000-fold selective for the other TrkA receptors, and if you calculate the ratio for the other TrkA-NAM molecules, you will see that it's much less. And obviously for the TrkA inhibitor, there's no selectivity whatsoever between the different TrkA receptors. And interestingly, if you look at tanezumab, which is extremely potent, it's 1 picomolar, the affinity at least for this antibody towards NGF, but it's not that selective actually. It's 1,000-fold selective towards TrkC, and there's been no reports on the selectivity towards TrkB. So actually the ACD137 molecule is the most selective molecule of all these different molecules targeting the TrkA TrkA-NAM or NGF-TrkA pathway. So I've been talking a little bit about BBB permeability and the importance of not targeting TrkB. So TrkB is intimately involved in memory performance or cognitive function. And here we have compared the TrkA inhibitor, larotrectinib, which is actually on the market now, in a model of memory in animals. And the higher the bar, the better the memory. And when we administer this TrkA inhibitor to these animals and then perform a memory test, we can see that there is a trend to decreased memory already at 3 mg/kg, and that's become significant at 30 mg/kg. On the other hand, when we administer the TrkA-NAM molecule, there is no effect of memory function, suggesting that we have, compared to TrkA inhibitors, at least a better side effect profile with respect to cognitive function. So summarizing this a little bit, selective and peripheral targeting of TrkA, we think that's a promising way forward for a new analgesics. We have identified highly potent negative allosteric modulators. We have synthesized, as far as we know, the most selective compound for TrkA over TrkB. And we think that this selectivity is really the key to obtain a safe compound with low side effects. But how about efficacy in animals? So we have tested it, like I mentioned, in several different models. This is an NGF-induced pain and inflammation model where we inject NGF. So it's a little bit artificial, but it's been used as our sort of screening models. So we inject NGF in the hind paw of the animal, and quite fast you will have an inflammation established within 10 to 15 minutes, and the animals will also have heat allodynia. And this is what we have tested to the left here, the analgesic effect of ACD137. You can see animals treated with only vehicle, they have a certain level of heat tolerance. This tolerance is decreased, so they withdraw their paw much faster when you have injected NGF, and you can see we can reverse this paw withdrawal latency in a nice dose-dependent manner with ACD137. And to the right, we also have the inflammatory effect of NGF. So you can see there is an increase on the inflammation. So that's actually the volume of the whole paw that we are measuring. And this we can reduce with ACD137. Going into a little bit, this is the inflammatory markers. Also, we have measured a couple of inflammatory markers through skin biopsies in these animals. And one marker that is really increased by NGF is CGRP, causing vasodilatation and thereby the resulting edema. This is increased a lot with NGF, and you can see we can block this increment with ACD137. And there is also a trend to an effect on IL-18 with NGF and ACD136. So we are not only reducing pain in this model, but we're also reducing biochemical markers of inflammation. So what about other disease-like models where pain is one component? This figure here shows the effects of ACD137 in a model of neuropathic pain, namely the chemotherapy-induced peripheral neuropathy, where pain is one component. So here we have used paclitaxel for inducing, which is a chemotherapy, and for inducing the neuropathy. And then we have administered ACD137 at 3 different doses and compared that to the effect of a known analgesics called gabapentin. And the higher the curve goes, the less pain the animals have. So here on the top we have naive animals, on the bottom we have in the black animals with peripheral neuropathy or painful peripheral neuropathy. And we can see at increasing doses that ACD137 can reduce this pain as much as 100 mg/kg of gabapentin, which is a quite high dose actually. And another model, this is a model of post-operative pain model, which is mostly a nociceptive pain model. So the animals have an incision, it's called the Brennan model, and then the pain is measured 24 hours after surgery. The same here, the higher the curve goes, the less pain the animals have. And you can see here that also there is a nice dose-dependent decrease of pain with ACD137 reaching, at least at 3 hours time point, reaching up to the same analgesic effect as tramadol, which is an opioid-based analgesics. So in these two models, both in a neuropathic pain model and in a nociceptive pain model, ACD137 gives a good analgesic effect. So we have also tested this analgesic effect in a model of OA, namely the MIA-induced model of arthritis in rats. So MIA, or monoiodoacetate, is injected into the knee joint of the animals, and after a couple of days there is an inflammation formed, and that will eventually lead to degeneration of the knee joint and a painful knee. So here we have administered ACD137 twice daily orally from day 3 to day 21 when we terminated the study. So here in this figure we have assessed pain by two different methods. Non-evoked pain, and that's measured on how much weight the animals put on each leg. And you can see here on the upper left of this figure is naive animals, and then they are given this monoiodoacetate, and that will change how much the weight they put on that particular knee or leg. So it's less weight. And here we can see that ACD137 is reducing that, so they distribute the weight more evenly. And interestingly, we have here compared the effects of ACD137 to tanezumab. It's a quite high dose given to these animals. And you can see that ACD137 is equally effective as the anti-NGF antibody tanezumab on non-evoked pain. And to the right, we have evoked pain. And you can see there's also a nice dose-dependent effect of ACD137 reaching up to the same analgesic effects as tanezumab, although it's a little bit hard to see. Tanezumab is the open circle on the top here. So essentially, both ACD137 and tanezumab more or less completely reduce the pain of these animals. So just wanted to say that this effect of ACD137 are as efficacious as the tanezumab anti-NGF antibody. But looking into a little bit more on the inflammation and also the joint pathology of these animals. So to the left, we have the inflammation of the knee joint. So it's measured actually by just measuring the diameter of the knee joint. So there's a quite pronounced swelling of the knee joint after administering this monoiodoacetate, as you can see here in the left part of the figure. And that is decreased in a dose-dependent manner with ACD137. And here tanezumab actually shows a little bit better anti-inflammatory effect with respect to the knee joint. We have also looked into the knee joint histology and tried to score the pathological events occurring in these knee joints. And you can see that the control animals here, of course, they have a perfectly healthy knee joint. Inducing arthritis in these animals will increase pathological condition in the knee joint, and that can be reduced significantly by ACD137. But this reduction was not statistically significant with tanezumab, maybe suggesting that targeting the NGF TrkA pathway with a negative allosteric modulator could be more protective for the knee joint than targeting NGF. We looked a little bit more into that, and I will just show you this slide as a background to how the knee joint cellular morphology or anatomy of the knee joint. So this is a healthy knee joint to the left. The artery cartilage is built up of different layers. We have collagen fibers, both vertical and horizontal, and then different cells in different layers before the actual bone here. And to the right, we have a figure depicting what it looks like in a arthritic knee joint. You have swelling of the knee joint, less synovial fluid, and the cartilage will be affected. You will not have this smooth surface, but more a ruffled surface. The collagen fibers will be disoriented, and also the cellular layers are not so well defined. So there are several abnormalities here in this knee joint. And then we looked with hematoxylin and eosin staining of the knee joint of these animals that we had in the study. So this is just one figure from several, but wanted to at least give you a flavor of how it can look on a cellular level in the knee joint. So to the left, we have a healthy knee joint from naive animals. You can see the cartilage surface is really smooth. You have a well-defined cellular layer before the bone. Giving or administering MIA or monoiodacet to these animals will give a ruffled surface. You see, you will not have this well-defined cellular layer like we have in the healthy joint. And administering ACD137 seems to be protective. You have a smooth surface, only very, very small ruffles up here, well-defined cellular layers here, and that is not seen with tanezumab. So this it almost looks like non-treated MIA animals. You have ruffled surface, cellular abnormalities. So we think here that ACD137 or other molecules targeting TrkA in this same mechanism will have, at least in these models, have a protective effect on degenerative processes in the knee joint. So clearly an advantage over anti-NGF antibodies, I would say. And just to summarize this, we have identified ACD137 through an internally medicinal chemistry program, highly potent and selective negative allosteric modulators, similar analgesic efficacy as the anti-NGF antibodies, and ACD137 is currently in preclinical development. And with that, I thank you very much for your attention.